It gives me immense pleasure to introduce Niamh MacSweeney to you all. She's a third year PhD student at the University of Edinburgh. And like many of the other studies here and elsewhere, students, I should say, she's had tremendous difficulties with collecting data over the pandemic. So she spent a long year getting ethics and getting all the data collection and biosampling in place, and was due to kick off mid-March, only to have the pandemic come and change her plans. Anyway, she is extremely resourceful and has coped very well, and she's an absolute pleasure to supervise. So I'll just hand over to her, and she's going to talk about biopsychosocial risk and resilience factors for adolescent depression. Over to you, Niamh.
Okay, great. Yeah, so as Heather said, my name is Niamh. I'm a third year PhD student. Oh, sorry. My computer. Yeah, okay. I'm a third year PhD student at the University of Edinburgh, and today I'm going to be chatting about my work on adolescent depression. So, adolescent depression, we know that adolescence is the time of greatest risk to develop depression across the life course. And in fact, half of depression cases emerge before the age of 25. And depression is actually the leading cause of illness and disability in young people. And unfortunately, depression in adolescence is associated with a more severe illness course. So what I mean by this is greater symptom severity as well as a greater number of episodes. And unfortunately, treatment efficacy for adolescent depression is modest at best. So tracing the roots of depression in the developing brain really is at the heart of my PhD, with the hope of better understanding why depressive symptoms have a tendency to emerge during adolescence. And to answer this overarching aim, I'm taking a threefold approach. So looking at biological factors, psychological factors, as well as social factors. And I'll discuss these in turn and during my presentation. But before I go into the work I've been doing over the past year, as Heather mentioned, there was quite a bit of upheaval to my PhD work over the past year. And I often use the metaphor of a whirlwind when talking about adolescence, because it really is this time of immense change in terms of our biology, our psychology, and our social world as well. And I feel like this was quite an apt description of my past year. So I had just received my NHS ethics application to launch a big data collection based project just after actually the Scholars' Day last year when COVID-19 hit. And unfortunately, this resulted in a suspension of data collection from March to October last year. So obviously this threw quite a significant spanner in the works. But luckily I had immense support from my supervisors and from my lab. And I was actually able to quite easily transition towards a computational based PhD. And what I mean by this is, rather than collecting my own data primarily, I was going to be using pre-existing data sets to answer my original research questions. And this actually resulted in a move over to the Division of Psychiatry, given the new focus in my PhD. But I'm hopeful that there are smoother times ahead, and I'm very lucky that I can use these big data sets to answer my original research questions. And these data sets also have the added scope of looking at other factors such as neuroimaging. And this is a very keen interest of mine and something that I'm really grateful that I was able to look at further. I had to quickly learn how to code, so that was a very steep learning curve, but yeah, I stuck at it and I feel a bit more confident now. And again, working with larger data sets with thousands of young people definitely allows to do more advanced analysis methods. So today I'm going to have a chat about my computational projects, as well as another data collection-based project that I've started recently. But firstly, looking at the computational-based projects. So when I'm talking about computational work, I'm using this wonderful study in the US called the Adolescent Brain and Cognitive Development Study, or the ABCD study. And this is a large-scale longitudinal study that was launched in 2016. And they've recruited over 11,000 young people aged 9 to 11. And the study is going to follow these young people across adolescence. And it really is such a remarkable I suppose, development in our understanding of, how young people develop over adolescence, especially in terms of brain development. So I'm really excited to get stuck into this. So I'm going to be talking about two projects that I've been using the or been working with the ABCD data set, and I'll just kind of talk briefly about these. So the first one that I did, and this was straight after data collections stopped. So I had the kind of, immense privilege of working with a wonderful postdoc in our lab called Shen, and we kind of led this project together. And what we were interested in is looking at brain structure and depression. So what I mean by brain structure is kind of cortical measures, so like gray matter in the brain, as well as white matter. So white matter is kind of the tissue that kind of connects different parts of the brain. And they're often found in these bundles or tracks. So when I refer to kind of white matter tracks, that's what I'm referring to. And we used the ABCD study and looked at nearly 10,000 young people to identify what are the brain features associated with depression in adolescents. And because to date, there has been quite a lot of research on kind of brain structural associations in adult depression, but we really don't understand that much about like why the kind of early emergence of these imaging features. So the beauty of this study is that we had like a huge sample size to kind of probe these neuroimaging associations. And I'm just going to kind of talk about the main results. So we looked at quite a lot. So first of all, we were interested to see, can we see similar kind of brain features in adult and adolescent depression? And indeed we can. So in both adults and adolescents with depression, we see a reduction in cortical volume, as well as a reduction in something called fractional anisotropy, which is a measure of the kind of integrity of these white matter tracks. So we see a reduction in those two measures, which might suggest that actually these features of depression might be present early in the disease course. But notably, we also see some differences. So we saw a reduction in surface area of the kind of whole brain, but then also kind of in brain regions individually. And this is something that isn't commonly reported in adult depression. So this may suggest that there is kind of some adolescent-specific vulnerability. Notably, we also saw stronger associations between caregiver reports of depression and brain structure. So across the board, caregiver reports of depression were more strongly associated with these brain features. And these discrepancies in reporting were driven by socio-environmental factors. So greater discrepancy was associated with things like family conflict and neighborhood safety. And this has definitely kind of laid a strong foundation to kind of probe these associations further in some follow-on work. But if you're interested in hearing more about the study, I feel like that was a bit of a whirlwind of what we looked at. You can have a look at our preprint there. It's currently under review. So moving on to the next computational project that I looked at. So I was interested, I'm kind of following on from the other study, like what are the other factors that might influence risk for depression during adolescence, especially in terms of, how this might relate to the brain features that we see. So the next project is looking at how does brain structure mediate or influence the association between early pubertal timing and depression, because we know that early pubertal timing is associated with a greater risk of depression. And I've taken kind of a registered report approach with this. So this is kind of an open research means of kind of publishing manuscripts, which involves 2 stages of peer review, and is quite like a transparent way of doing science. So I'm using both physical and hormonal measures of puberty, so looking at kind of, testosterone, estradiol, and how this might indicate risk for depression. And then I'm interested to see whether or not these kind of the measures of puberty and the measures of depression are kind of, or whether this association is mediated by brain structures. So I'm kind of taking like this, so there's 2 stages to this project at the moment. I'm doing some pilot analysis looking at the association A and B on the diagram, and then in my kind of confirmatory analysis, I'm going to look at whether or not there is kind of a mediation effect in kind of pathway C, as you can see on the diagram. And this will kind of involve 2 time points from the ABCD data set to test whether mediation has occurred. And, this was quite a steep learning curve in terms of like working with these huge data sets. So I've put all of my scripts on GitHub. If anyone ever finds themselves QCing or quality controlling hormone data, definitely get in touch. So then moving on to my data collection project. So luckily, data collection was allowed again in November. And this, meant that we could start a study that we were meant to kind of start back last year. So this is a neuroimaging study. So we're using something called a functional MRI to kind of better understand what's going on in the brain when young people are irritable. And this was a kind of co-produced youth researcher design. So we actually worked with young people from the very outset, which has been a fantastic way and kind of, fantastic experience and really enjoyable for everyone involved. So we know that irritability is one of the core symptoms of depression. And, understanding what it is about irritability that's kind of linked to depression may kind of improve our understanding of the early origins of depression, as well as its future trajectories. And irritability hasn't really been studied that much, especially in an imaging context. So the aim of this study was to kind of develop a fMRI task. So this is a task that young people will do in the scanner, and we're kind of interested to see what's happening in their brain when they're in an irritated state. So there were kind of two stages to this, and both involved young people. So last summer, we worked with a wonderful student called Simil as part of the Nuffield Researchers kind of summer project. And Simil helped us kind of develop the task that we eventually ended up using in the scanner. So we asked young people to come up with scenarios that they found irritating. And then these were compiled into an online survey and rated by a separate group of young people. And then we chose the top 20 scenarios for use in the scanner. So some of the examples included, like, I find irritating when I'm talking to someone and they're being rude to me, or when people tell me to do something even though I was going to do it. So I think they're quite like generalizable. I feel like, they're all quite irritating, these scenarios, regardless of age. And then this then kind of formed the basis for the phase 2 of this project, which was launched in November. So thankfully we were able to recruit young people again. So I spent a lot of time over the past couple of weeks at the scanner recruiting young people with depressive symptoms through schools and universities. And although recruitment has been quite a challenge, we were able to recruit 30 people, and we've recruited and scanned 30 people, and will hopefully be finished data collection in May. And this is kind of a pilot study that kind of, will test whether or not kind of this kind of task is feasible on a large scale. And young people have been and will continue to be involved in this project. And we've actually co-authored an abstract for the MQ Mental Health Summit next week. And they are named as, the young people are named as authors on that. So I suppose looking ahead, so my immediate plan is to finish data collection for the fMRI study and carry out data analysis. And then I'm also going to continue working with the computational-based puberty project and finish both of those up kind of over the summer and early autumn. And then, because I'm in my third year, I'm going to start my final 2 PhD projects, which are kind of informed by the existing computational work that I did, looking at things like contextual associations and cognitive factors. And then I suppose kind of aside from like my direct PhD work, I've also done some other things this year. Highlights, I think, have been like setting up an open research journal club at Edinburgh, which is really kind of, created this wonderful community about kind of transparent ways to do research. And then also a very good public engagement project that I took part in, called Your Depression, My Depression, which was creating digital stories to kind of communicate our research. But I just want to end by thanking my wonderful supervisors, as well as my collaborators over the past year, especially our 2 youth researchers who are a pleasure to work with. And thank you very much.